Oh, it does? Ah, got it. Got it.
Hey, yeah. Good afternoon, everyone. I'm Farnaz, a second-year PhD student in Dr. Eric Garrison lab. I'm honored to introduce Dr. Sudmant to you all. Dr. Sudmant is an assistant professor at the Department of Integrative Biology, UC Berkeley. He obtained his bachelor's in computer science and a PhD in genome science from the University of Washington. The Sudmant lab at UC Berkeley combines computational analysis, statistical approaches, and laboratory experiment to explore genetic variation and molecular characteristics in both whole organisms and individual cells. The research focuses on understanding the aging process while also investigating the evolution of genome organization and diversity of cell types. Let's give a warm welcome to Dr. Sudmant.
Thank you. I appreciate the invitation. I'm really happy to be here. I've never been to Memphis. It's a really beautiful city. And it's really nice to be amongst a bunch of friends. There's a lot of folks here who I know very well and have a really nice time. Collaborators, people I've done some really fun science with over the last decade or so. Really nice to see them and really nice to meet some new people too. And then I have to say, so this morning I met with all the students, the first and the second year students, and that's something that I've never seen in that particular form. They gave their little, everyone gave a little talk. I guess you know how this works, and it was awesome. It was just really fun to meet all these students doing cool science and to hear about, you know, their journeys and stuff. And I really enjoyed that. So it's a testament to the sort of the environment that you folks are creating here. It was really cool. All right, so I'm going to talk today not really about aging. I'm going to talk about genome structure. But I'll tell you briefly about some of the things we do in our lab. We study several different things. We study the evolution of aging. We study how throughout aging, different molecular processes become perturbed and how the fidelity of those processes is maintained or lack thereof. And we study the evolution of genome structure. So this work in aging is really fun and really cool, and it's sort of a cheat code and that just like sort of lets our lab do goofy projects whenever we want. So a few years ago, we looked at the genomes of all these fish, these rockfish that live off the coast of California. Some of them live for 10 years, some of them live for 200 years. And so, you know, we were able to identify some of the genetic drivers of lifespan in those fish. And more recently, we've been exploring other systems, so like bats. So this is some work we have in bat genomes, which is really exciting. It's on bioRxiv right now. And bats are really cool for a lot of reasons. But one of the main reasons is that, so if you, this picture here on the left, the x-axis is log mass and the y-axis is log of lifespan, maximum lifespan. And this is all mammals, or a lot of mammals, not all mammals, but mammals for which we know their lifespans. And you can see that there's a very strong straight line, right? So bigger things live longer. So, you know, whales live longer than elephants, live longer than people, live longer than horses, live longer than dogs, than mice, right? But bats, as you can see, kind of kick this trend, which is really cool to see. And so, you know, one idea that we have is that this we call it non-allometric lifespan scaling is maybe being driven by specific molecular pathways and genes. And so that's why we're studying those. And so the postdoc who just is about to leave for his own faculty job, Juan Manuel Vasquez, goes out in the field, catches bats, takes wing punches. We make T-to-T genomes from them, and then grow up these cells in a dish and then do experiments on them, which is super cool. So this sort of a second half of the lab is sort of thinking about this aging process a little bit more in depth and try to thinking about molecular processes and quantifying them through age and lifespan. And I'll just quickly mention this work on the right here. And I won't go too much into this, but in this project, we used this type of sequencing called duplex sequencing, which allows us to look at somatic mutations and we specifically did this in mitochondria. And we tried to look at how somatic mutations accumulate in mitochondria throughout lifespan of these mice. And there was a lot in there that I won't go into in depth, but I just want to show you one tiny little thing. Okay, so on the top here, this is a mitochondrial genome that's been unrolled. Okay, And all those little dots up there, it looks like a Manhattan plot. That's the mutation frequency in a mouse. So there's all these mutations happening at like very, very low frequency in mitochondrial genomes as they're replicating. And this picture here is just really simply showing, the old mice have more mutations than the young mice. Fine. But here's the really interesting thing is that when you look at the types of mutations that exist, they're actually distinct in mice from primates. And so this is a picture showing the different kinds of mutations that we observe, and we've normalized everything to C to T mutations. So that's normalized to 1. And so what we can see here is the second most common mutation type is G to T, or the signature of oxidative damage. We saw it in our study, and when we look at other studies as well, we can replicate the signature. It's the second most common signature. But this is completely different from what you see in humans and in macaques. So primates do not have this oxidative damage signature. So that's really interesting. The fact is that, as you're getting older, your DNA is getting damaged, but it's getting damaged in different ways for different clades. So why is that? It could be because there's just more oxidative damage in primates. Could be that there's better oxidative damage repair in primates. Could be that there's better, cleaning up of things in primates. We don't know, but we're exploring this, and we have a project on the go, which is trying to compare these somatic mutational processes across as many primates as we possibly can. And this is, again, work that I'm not going to talk about, but I think it's pretty exciting. And I will come back to this mutational processes thing later, so I wanted to highlight it. And then I also want to, I'll just also note one other thing. So as I go along here, there's two things. So one, this said 12 to 130.
You can take this. Usually people present for a while and we talk.
Because we interrupt a lot.
Yeah.
That was the second thing I was going to say, is that I cannot abide a single interruption. No, I'm just kidding. Please, no, is that please, yeah, give me feedback as you go. If you have questions or something's not clear, yeah, let's just talk about it. Yeah. So what I wanted to talk about today was 3 incomplete things, because that's what this is about, is talking about stuff that's not ready, totally ready for the world and what's exciting and new. And I'm really excited right now about structural variation. So I want to talk a little bit about some primate genomes we're building, and this really exciting time we're at. I know a lot of folks here are really into T to T genomes and pan genomes. I don't have to sell that, I don't think. I want to talk about how we can extend that from primates, looking across the entire clade of vertebrates, which has been really exciting. And then we'll return to this question of mutational profiles. And then finally, I have just this tiny snapshot of this AI project that we have right now, which is kind of blowing my mind, and I don't fully understand it, but I'm really excited about it. And so that's what I'm gonna try to tell you all about. So who here knows what a structural variant is? Okay, I'll skip this slide, right? So structural variants are big things, right? Yeah, this is for Andrea. So this is a structural variant, right? Conversions, deletions, and duplications, right? Things that are not SNPs, right? Why do you want to study structural variants, right? There's a lot of reasons to study structural variants, but I want to give an example that is hopefully dear to Andrea and Eric's hearts in this super cool paper we published collaboratively very recently on amylase. And so one of the first reasons to study structural variants is they have extraordinary allelic diversity, right? So SNPs, well, actually, I guess you found a lot of multi-allelic SNPs in your time, but in general, we usually think of SNPs as biallelic, but structural variants can have extraordinary numbers of alleles. And so this is a case from this paper we published. Each of these represents a different structure that we observed at this locus called the amylase locus in humans. And so all of these are ranging in size by, they vary in size by hundreds of kb, and they're all very different. So the first reason to study structural variants is a lot of diversity, right? Second reason is super fast mutation rates. So from this same project, I won't get into the details, but we were able to basically use the fact that we have linked haplotypes linked to the structures next door to date when those structures emerged and figure out what the mutation rate is of a locus like this. And the mutation rate for this particular gene, amylase 1, for gains or losses, is 2 times 10 to the negative 4, which might be kind of a meaning per generation, which maybe is a meaningless number, but to most of you, but unless you're familiar with these numbers, what it means is that this region is mutating at a rate that's about 10,000 times the SNP mutation rate. So mutation is happening really, really, really fast. There's a lot of stuff happening. There's a lot of material being generated really rapidly. And then finally, structural variation breaks our models. It breaks stuff. And so one of the most important things in all of genetics is linkage, and what linkage enables us to do and to study, right? So we've used it in pedigree studies for a very long time. And of course, that linkage drives correlations in allele frequencies, which we can use in GWAS to identify associations between phenotypes and the genome. But what we found, and what perhaps is not surprising, is that these structurally variable regions, they actually mutate very, very, very rapidly while maintaining LD. And what that means is that flanking SNPs don't tell you what's in the middle, right? You cannot genotype a structural variant necessarily or very well from the flanking SNPs. And the reason for that is the mutational process. So people who are sort of familiar with this and know about how structural variants occur through non-allelic homologous recombination, we usually think of it happening like this, with two homologs, right? And these two homologs mispair. And when that mispairing results in one homolog that has more copies and one that has less copies. And that does happen, but that will disrupt LD. So you should be able to see a signature of that in linked SNPs. GWAS should work. Turns out this is actually rare. And this is not how non-allelic homologous recombination usually happens. What it often happens with is between sister chromatids. So when it happens between sister chromatids, you don't mess up LD at all. You have perfect LD because the sister chromatids are identical, or even intrachromatidally, right? And to me, this was kind of mind-blowing. And then, you know, I went and I read some yeast textbooks, and of course they had this well figured out many, many decades ago. They knew about it. And of course, thinking about it a little bit more carefully, if you look at the Y chromosome, a huge proportion of azoospermias are caused by deletions on the Y chromosome through non-allelic homologous recombination events. And how are those happening? Well, they cannot possibly be happening between homolog NAHR, because there is no homolog, right? So everything sort of fell into place. But the point being that these are like super cool, super fast evolving regions to study, okay? So the other reason why structural variants are pretty cool is kind of just because of the time we are at right now, this particular golden era. And so what I have here plotted is sort of my timeline along the top. I was born before 2000, but the red is back when I lived in Canada. And then in 2008, I moved to the United States. And in 2018, I started my faculty job. And if you look at just what's happened, it's truly remarkable, right? We have seen, I was like, and you know, and many of you probably remember this, is that, you know, in 2008, we had the first short read sequence genome. And that was the first month that I started my PhD. And by the time I finished my PhD, Eric and I were publishing on thousands of human genomes. So this huge sea change happened, which is remarkable. Along the way, of course, we got the Neanderthals and Denisovans. But the thing about all of this, everything that happened over this time period, as well as an enormous amount of progress in human health and you know, discovery, disease, companies, you know, like 23andMe and identifying the causes of huge numbers of human diseases. The thing that was kind of remarkable about all this is that all of this was lying on the back of the human genome. Everything required a reference genome. Everything is basically entirely dependent. Every RNA-seq, every single you know, sequencing experiment, every kind of omics you could ever do, or potentially the mouse genome, or potentially the rat genome, but it's reliable on these reference genomes. And that, of course, as you're all aware, has completely changed, right? We have now T to T genomes. We have these incredible resources, and we actually don't just have a human T to T genome, we have pan genomes. We have genomes from humans that are at better quality than the original human genome, and we have both haplotypes. And the thing that's also been happening that's pretty remarkable is we're getting this from all kinds of organisms across the planet. So the three stories I'm going to tell you about today are sort of leveraging all this cool new data we can do and figuring out what we can learn and what we're going to be learning about that we could never see before. All right. So these projects are fantastic and phenomenal, and you folks, many of you have really exciting projects involving mice and rats. But one of the things, and so you'll appreciate that one of the things that's of course very important about human genetics is when we're thinking about it, we need to put it in context. And one of the important places to put it in context is in evolutionary context. And so one of the things we've been doing is building a complement to the human pan-genome with chimpanzees. So these chimpanzees, and we're basically making complete haplotype resolved reference assemblies from chimpanzees, and we call this the pan-pan genome, because this is the pan genus, right? So here's everything that we have now for humans, which is remarkable, right? We've got about 200 humans, and there's our little slice. We have 29 chimpanzees that we've done massive sequencing of. And I'm not sure if people are familiar with, but chimpanzees are quite different from humans. So chimpanzees split from this sister species, bonobos, about 2 million years ago, and they had these 4 different subspecies that are all have quite deep coalescent lines, maybe 160 to 250 to 500,000 years ago. So these are very, very different. And so we've made genomes from all of these, and I'll just tell you some very quick things. This is very much work in progress, but I'm really excited about it. So we did it, yeah.
Not the bonobos.
We did it from bonobos too. Yeah,
Okay.
yeah. So we've got, well, you'll see here, we've got 15 Western chimpanzees, 4 hybrids, 3 Eastern, 2 Central, and 5 bonobos. Yeah, yeah, which is cool.
There needs to be a better word for, I guess, bonobos and chimps, or do you just consider it considered?
So bonobos originally, one of the names for them is pygmy chimpanzee. So, the word chimpanzee is used sort of as a pan term, but it's a good point. Yeah, yeah. So, all of you know what this kind of data means. So for all of these genomes, we've done hi-fi sequencing. And so these are, we assemble these haplotypes for these, all of these. So there's 3 different sort of flavors we've done. Some of these genomes we've done just with hi-fi, some of them we've done with hi-fi and hi-C, and some of them we've tried to go all in T to T on them. And so this is, the N50 sequence length. Of course, you throw more stuff in, it gets better, but it's sort of gives you a sense of what the quality of this data is, right? It's not only a small proportion of it is going to be T to T level, but it's still extremely powerful, right? And what's really cool is then you can start to integrate this with all the extant short read sequencing data. And this is actually something that's surprisingly difficult to do. But on the left here, so this is a PCA. This PCA is done with individual haplotypes. So these are from the long read genomes. And so you can see the different species, subspecies clustering together here. And then here is when we integrate long read and short read data, which is actually, there's surprisingly difficult and fiddly, but it works really well. You get all the right clustering coming together. And so one thing that's kind of interesting when you start looking at these genomes and you're trying to sort of interpret, what do you expect to see, is if you start looking at diversity statistics. So here these are a whole bunch of different chimpanzees, all the different chimpanzees we've looked at here, and the y-axis is heterozygosity. So that's the number of variants that differ between the 2 chromosomes. And the dotted lines here tell you what sort of the average heterozygosity is for an African and a non-African human. So what you'll see is that basically these chimpanzees are way more diverse than your average human. And that's pretty cool and pretty exciting. And what we can then do is we can make a plot like this. So we're looking at structural variants here, and we genotype, we identify structural variants in these chimpanzees. And then you add, you put a bar for your first chimpanzee, you discover 20,000 structural variants, then you do it in the next chimpanzee, and this is the added number of structural variants you get. So this curve is the total number of structural variants. And you do that exact same analysis in humans using all the same things, and it looks like this, right? So you're getting way more data. Yes?
When you compare the 8 or 9 billion of human population, what could be your explanation? How much
So if you match the same number of individuals.
9 billion people based on the earth, and we had that kind of variation, but number of chimpanzees probably a lot of.
It's smaller, it's much smaller, yeah. So why does that happen? So the reason why our population is so much less diverse is because, we traditionally have actually had a much smaller population size, right? There's many fewer of us. And so basically this explosive population size is only actually within the last 10,000 years or so. I mean, even probably more like the last few hundred years, actually. So that's why you don't see as much diversity. It hasn't had enough time. Whereas in contrast, chimpanzees had this huge population size compared quite a, maybe 2 million years ago. And so that's what we're seeing. And these are fairly old variants. Yeah. Sorry? Exactly. It's about the history. Yes. Yeah, exactly. Yeah, that's a good question. All right. So now we can start to compare these and we can look at what's different. And so this is, I think, super cool. All right. So this is a plot of structural variants in humans and chimps. So this is the size distribution and count. So just a histogram of insertions relative to CHM13 and deletions relative to CHM13 in humans. And so what you can see here is that, there's more of these small things. There's this huge peak here, and then it goes down. So really big things are quite rare. And these peaks, which I colored in here, are mobile element insertions. And so it's well known that the human lineage has like a high activity of ALU mobile elements. That's what that big peak is there. But what's really interesting is then you compare this peak to the one from chimpanzees, and the bonobo one looks very similar. And so the way to look at this is compare the size of these colored peaks to the size of this sort of density down here. The ALU peak is much smaller, but the SVA and L1 peak are much higher. So what does that tell us? It tells us that these mobile elements have different activities in different species. So humans have very high activity of ALUs, whereas these chimpanzees, we now know, we are able to see here that they seem to have much higher activity of SVAs and L1s. And you can quantify this in a couple different ways. On the left and the right, it doesn't really matter here, but that's being quantified by this theta or pi metric here. So signs has the most activity in humans, blue is the highest, the lowest activity in bonobos. Lines have the highest activity in chimpanzees, followed by humans. And then these SVA retrotranspose ones also have like the highest activity in chimpanzees.
Is that pi-wise diversity within ALUs in bonobos, for instance?
Yes.
That's what we're looking at.
Yes.
Pi-wise within the ALU within bonobos.
It's within the population.
Within the population.
Yeah. So what we would ideally want, right? Ideally, you would want to have a mutation rate. You want to know what is the number of new L1 insertions per live birth, or what have you. But that also, it's very hard to get that, and also that could have changed over the last. So basically, this is a statistic that tells us how active these are and how active these have been in the population since, in the population over the last, say, million years or so. So there's other cool stuff that are really cool reasons to study chimps. One of them is that it turns out, so our species is susceptible to many of the same insults that chimpanzees are susceptible to. So malaria, of course, has plagued our species for a very long time, and malaria is caused by Plasmodium falciparum. And the malaria parasite enters human red blood cells by attaching to these glycophorin cell surface blood receptors. So that's what's being shown right here. And several years ago, this really cool study was shown that, so these glycophorin genes here, which are shown there, there's a specific haplotype where there's a duplication of these glycophorin genes, creating something called, creating a 2 gene fusions, and this is called the DAN2 haplotype. And this DAN2 haplotype actually changes the red blood cell surface tension. And by changing the red blood cell surface tension, it prevents Plasmodium falciparum from entering. So this particular haplotype is at very high frequency in sub-Saharan Africa. And so we started looking at this, 1,000 genomes gene read depth. We started seeing, look at all these, in the human region, we have all these deletions and duplications, that's cool. But then we started looking at chimps, and this same locus, there's all this stuff going on here. And so what's happening here? Well, this is likely a case of convergent evolution, or potentially ancient balancing selection, which would be even cooler. So basically, huge amounts of diversity that have been maintained over time due to balancing selection. So if we, here's what, whoopsies, we're going crazy here. So here's what these structures look, here's what the human structures that have been identified look like. This is from array data. So, you know, these are indicating, I think, duplications and deletions, a lot of variation at this locus. But this is from looking across a huge number of African individuals. I think there was like 20,000 individuals in this study. So we sequenced 30 chimpanzees. That was all we sequenced. And this is what the structures look like in chimpanzees. So this is a pangene graph. A pangene graph is a kind of a nice, cute way to start looking at a locus fast. And basically what it's showing is it's showing the structure of a locus, but only the genes. And so that's the graph on top. And we were able to find, I think 12, I think there's 12 there. In these 30 chimps, 12 different haplotypes, this extraordinary haplotype diversity. And so these genes here, so FREM3 is on one side, HHIP is on the other, and everything in between are GYP genes. So the one, like this one, for instance, is labeled, you know, lock 46, 15, 20, but that's just because it's a GYP gene. These are all GYP genes inside here that they just have never been seen before. So this insane structural variation at this locus, kind of like that amylase story I showed you before. And I just think it's so cool that we can start to dig into these. And yeah.
Balance and selection should be able to do. But how can you think about empowerment mind? How can you do that with such a complexity of data? You know, how it's not just simply looking at mutation rate and scaling back in time. Do you have any idea?
How do we just identify if it's convergence or or
If you'll be able to date the duplication, then you can answer this question potentially. And so the question for you is like, if you have thought of a way of dating the duplications in perpetuity.
Yes, so we do, and we're working on it, but it's not done yet. And the way we're doing it is, so one of the things, I'm not really talking about, one of the things that's just so exciting about these long read sequences is that, you know, we used to spend, and we still do, inordinate amounts of CPU cycles statistically phasing human genomes. And when you do with these genome assemblies, the phase just falls out. You have a fully phased assembly. So because you have that phase, you can do these really nice population genetics analyses. One of them you can do is do an ancestral recombination graph reconstruction. And so we're doing that. We're doing ancestral recombination graph reconstructions, which we kind of sort of did in the amylase as well, across this locus. And what we should see is that what that does is that it measures the TMRCA, the time of most recent common ancestor, along the genome. And when you do this for like MHC, it's like crazy high. Like there's regions at MHC that are like 30 million years or more TMRCA. And my hope is that this will be like, you know, super old. I don't know yet. I'm hoping. We'll see. Yeah, that's a good question. Yeah. Yeah. So that's like our little nugget of this story, right? This is this pan genome, and several folks here involved. I probably forgot somebody, and I apologize, but very grateful to the UTHSC crew for their assistance, as well as, you know, Italians, either all around the world. Yeah. So this is thinking about this in chimpanzees. We also have another project also involving some of the great folks here, trying to sort of extend the lineage and build T to T genomes from the rest of the primate lineage, which surprisingly is, you know, there are very few T to T or high quality genomes from this enormous amount of diversity across primates. So there's about 400 extant primate species and only about a dozen or so primate genomes, and that's highly biased towards the great apes, and even like very important medically relevant macaque species and African green monkey. They have like really crappy genomes. They don't have very good genomes at all. So with this project, we're basically going crazy. We're doing as much sequencing as we possibly can, and we have 10 genomes complete. Thank you to I think so. The one person who's not, I think Shuo, I didn't put you on here. Where's Shuo? I apologize, you're not on the slide, but Shuo has been helping a great deal with this so far. And I'll just show you like a very quick snapshot of what this looks like. So, I mean, this is kind of silly, but I actually really love these figures. So this is just an ideogram showing the chromosomes. And the little black lines indicate gaps in the chromosomes, but these are the homologs here, right? So you can see most of the, and then these parts, these little triangles are the telomeres, and these orange things are the centromeres. So you can see that, like, you know, we basically have assembled completely this olive baboon, both haplotypes. And you know, there's some, you know, stats here, you know, QBs and boostos are all like very, very high. But it's much more fun to just look at this. You actually have the complete genomes, right? And then even just like we like literally just got these genomes in the last 2 weeks or so, or maybe, and these pictures are actually, we have the centromeres like this week. So that's why, so I just added, or they just got added here. But now you can start to see really crazy things happening. So these centromeres here, they're pretty big, right? They're pretty hefty centromeres. Those are the orange blocks. Now let's look at this amazing guy, the white-faced Saki. And look at these tiny, tiny little centromeres. It's got tiny, tiny little centromeres in here. And there's also huge variation between the homologs and the centromeres, right? So this is another part of the genome that, like, we never were able to look at before, right? We could never look at centromeres really until these T2T genomes came along, which is, yes, they're all about the same. Yeah. No, the number of chromosomes is different. Yeah, so this one is really cool. So look, it's okay. So this one has 20. This one has 20, well, you know, 20 autosomes. This is 23 autosomes, right? And then check out that this one is 24. Okay, I don't know why I'm showing this one. This is just a nice one, cute little monkey, whatever. And then this one has 31. All right, so this woolly monkey. So the next phase of this project is then to start taking these chromosomes and saying, well, where did you know, how did they emerge? How do you get, like, what? Yeah.
So question, but that's, you know, the number because you've done cytogenetics in this.
No, we know that
By this.
just by the assembly. It's amazing. Yeah.
I mean, you have the telomeres on both ends of all the scaffold.
Yeah. So I think you can see here.
It looks like acrocentrics that don't have.
These ones don't have, no, I don't, well, I don't
You
think
know,
they're acrocentrics because then I don't think we would get the telomeres. Well, I guess they could if they had tiny telomeres, tiny centromeres.
Really not expect any inversions in one individual in any of these.
We haven't, I'm not showing the inversions. There's definitely inversions. These are not.
I mean, big, like 10, 50 megabase inversions.
There are some, they're not, you can't see, I have one here. You can't see them. But there are a couple. There's, I think it's in one of the lemurs. There's like a massive, like 15 megabase inversion on one of the haplotypes. And it looks definitely real because we can.
We're doing a simple cross and we have 10 inversions, all between 10 and 50 megabases in mice.
Yeah, I mean, so mice are going to be different, right? Because, well, what's exactly driving those could be different, right? It could be like, so I don't know what species it is, but in like some of the Paramiscus have like insane.
No, this is all
Yeah, it's all muscle. Yeah, that's wild, right?
laboratory audit.
Yeah. Anyway, so I just think this is super cool what we can start to do now. Yeah, and so that's like that's all I'm going to talk about primates for now. If anyone has any other questions about these guys, but I just am super stoked. Yeah.
Sort of odd question. So you showed basically transposons, and I'm thinking of like prey predator, things that regulate transposons. Do they behave in some sort of prey predator behavior?
Yeah, they absolutely do, right? So the other word for that is the Red Queen hypothesis, right? Is that you have to keep running as fast as you can to stay in one place. So basically, what it says is the transposons have to out-evolve the things that are silencing them, and the things that are silencing them are still, so they're going at the same rate. Yeah. It's a good thing to look at. We should look at it for sure. I'm trying to think if we can, so I want to look at that. It's a really tricky thing to like what you would ideally want to do is identify the novel genes that are involved in silencing lineage specific transposons. And let me continue because I'm going to talk more about transposons. And I think not a better way, but a more and potentially easier way to look at it is to look over the whole clade, vertebrate clade, which I think would be really cool. Yeah. Cool. So like, okay, we could do all this amazing stuff now, and we're making all of our own genomes for all of these primates, which is incredibly fun. But there's the rest of the world, right? There's the rest of animals which have enormous amount of untapped and unexplored genetic diversity. And I remember, I think Eric, you and I were probably both at this meeting. Biology Genomes, like 2011, maybe 2012, and Molly Pajorski presented this paper, which prima facie is kind of a little boring. Basically, this is a plot she made of the heterozygosity, or the amount of SNPs across as many species as you possibly could, going down here from lynxes, right, which are completely inbred, and then I think only a few up. Here's us, right, also reasonably inbred, all the way up to these incredible different mostly insects and things and aquatic organisms that have extraordinary amounts of heterozygosity. So just a lot of diversity between the pairs of haplotypes that exist. And so this is not that long ago. In 2012, we still didn't really know a ton about just sort of the breadth of diversity of organisms on this planet. And what's more, we have, even till pretty much now, no idea how this SNP diversity scales with structural diversity. But for the first time, we can look at this and we can explore this. And it's because we have these haplotype resolved genomes. And so what the assemblers remarkably, as sort of as it was pointing out, they just like pop out two haplotypes and they're like perfect. You have to do some cleanup of them a bit, but it works really well. And then what you can do is you can take those two haplotypes and you can just look at them against each other, and you can identify the SNPs, but also all these structures. And so this is segregating variation that's in the population. This is kind of like thinking about like the difference in transposons in the chimpanzees and humans, but now looking at just any species. And what species are we going to look at? Well, we've been basically sneaking around the internet, trying to download as many of these recently released massive scale genome assemblies as we can. So there's a huge amount of data from the Vertebrate Genome Project, from the Darwin Tree of Life, and from the California Genomics Project, the CCGP. And so to date, we have 499 diploid assemblies, though that will change. I think we just started downloading a whole bunch more. And this is what that data looks like. So it's shown in two forms here. This is just a pie chart showing the breakdown of the different clades, and here it is on the clade. So these are the jawless fish here, cartilaginous fish here, the rayfin fish in the green, amphibians in the purple, mammals, reptiles and birds. I guess reptiles and birds are reptiles, but you can split them up there, right? So that's everyone we have, and then you can start sort of looking at things like assembly stats. So how game ready are these? They're actually pretty good. So what this plot is showing is basically the N50 contig length of hap1 versus hap2. So on the larger one, we put on the top, and then there's a rainfall or a tear falling down to what the second haplotype is, right? So there's a couple things you can take home from this. So first of all, you can see the mammalian genomes are the biggest, right? They're the yellow. I guess the mammalian and the reptilian genomes are the biggest over on the left. Fish genomes tend to be the smallest. And in general, there's it looks worse than it is because you're only seeing the black, but there's 500 things here. And so most of these haplotypes are pretty good. Both of them are both very, very high quality.
It's a quality metric, not about the length of the genome.
Yes, yeah, this is mostly a quality metric.
Amphibians are largest than the mammalian.
The amphibians are not that big.
There's some in there.
Yeah.
10, I think there's an axolotl that's 25.
Yeah, and that's a good point.
But anyway.
No. So one of the problems is actually, so the amphibians, basically they break all the tools. So there's like 3 or 4 amphibians that we haven't been able to do anything with because, back, when repeat masker was written, I don't know,
Is it polyploid or?
Well, because the chromosome gets longer than the 32-bit integer.
Exactly. Yeah, exactly. Yeah. Yeah. So we've been doing that. We've been breaking them into pieces. which is just super annoying. Yeah. But this is just a quality metric. So now, okay, so that's cool. So then what we do is we take the two haplotypes and we compare them to each other and we identify structural variants and SNPs and things like that. So this is, and then we can go back to that analysis that Molly Pajorski did, and we can look at the distribution of genetic variation across the world. So this is heterozygosity. So the average number of SNPs the rate of SNPs between the two haplotypes here. And it's, here's one base pair indels and SNPs here. And what you see is, there's a huge range in heterozygosity across species. And there's more variation within than between species. It's not like, I mean, the cartilaginous fishes, there's a little bit less, but there's just not that many of those, right? It's not like there's certain clades are like way more diverse than other clades. But things get kind of more interesting when you start to do this for structural variants. So this is the SNP heterozygosity plot, and here's the structural variant heterozygosity plot. So here, this is inversions and duplications, and then insertions and deletions. And this is not weighted by base pair. Like if you have a structural variant, you just count one, right? And what you can see is this extraordinary result in that there are clade-specific patterns here, in that some clades, particularly amphibians and raced fishes, have a lot more structural variation, heterozygosity. So this is not, again, this is not like the genome. This is like segregating in the population, variation that's in the population. Yeah, Enza. It doesn't. I don't know if I have that slide. It's normalized for the size. Yes, it's normalized for the size. Yeah. So this is the rate. So this is like it's basically what the number is. It's the total number divided by the size. Yeah. And so you start. So basically you have more structural variants. Okay. So I'll come back to that. So what else is going on here? So other people have shown this, but this is a very simple thing you can do is you can take a look at all these different species and you can group them into those that are threatened, like IUCN threatened, or not threatened, right? Things that are like, and basically you see lower diversity in the threatened taxa, right? So here in the reptiles, for instance, lower in the red than in the blue, right? And that's the same across structural variants and SNPs. And this makes sense, right? So if you're a threatened species like a lynx and you're mating with your cousin all the time, you're just homogenizing, right? Okay, so that's. Is
that true even correcting for what the effective population size probably was 10,000 years ago? And so, a lot of these species are without any effective population size like we do.
Yeah. It's not correcting for that, and it's a very ugly analysis for that reason. It's not clean or elegant at all. You're absolutely right. It's basically just sort of showing. It's actually, to be frank, it's kind of unclear what it's showing, but I think what you're kind of getting, in the extreme scenario, right? You're showing, like cousin matings, right? And that's maybe what's pulling most of the things are down, but you're right. Cause like we now have a much larger population size, but yeah, it's basically skewing towards more recent things. Yeah. So what you can do, and what we did is we plotted these two rates. So the x-axis is the single nucleotide heterozygosity rate, and the y-axis is the structural variant heterozygosity rate. And they fall in a really nice straight line, which makes sense, right? The processes or the forces that are shaping genetic diversity in a population, they're acting, basically diversity is shaped by patterns in the population, demographics, and that's not going to change basically if SNP heterozygosity or structural variant heterozygosity. But the thing that's crazy here is that the slopes of these lines are different, right? And so what that means is that for a given amount of diversity at the SNP level between two chromosomes in an individual, there's going to be more or less structural variant diversity. And so we can quantify that by taking just the ratio of those. So we just divide structural variant heterozygosity by the SNP heterozygosity, which is again not perfect, but it's actually a pretty reasonable metric. And what that gives you is a picture that looks like this. which I think is mind-blowing. So what you're looking at here is the ratio of rate of structural variation, structural variant heterozygosity to SNP heterozygosity. And so what this is telling you is like how much of the diversity in the population is being driven by either of these two forms of genetic variation. And in all of the fish lineages, the cartilaginous fishes, jawless fishes, rayfin fishes, and amphibians, the vast majority of the diversity in the population is driven by structural variation. And it goes immediately, sharply gets down in mammal. It comes back up a little bit in the reptiles, and it's very, very, very low in the birds. So I think that's really cool because it's telling us this remarkable thing about like the world around us and what's shaping and patterning diversity is actually substantially different between clades. Yeah.
So do you think that the species tend to be larger compared to something?
I don't think so. I think because that should be reflected at the SNP level too, right? So I think so I'll show you what I think it is, and I think what's basically driving this.
So it's like, it's less really damaging.
Yes.
To have
Yeah.
way more alternatives, see what I mean?
Yeah. So, okay. Yeah. So great. Yeah. Enza has a great, really smart point here is that basically if your effective population size is larger, okay, or let's go back. Structural variants tend to be on average more deleterious than SNPs because they're big and they kill stuff, right, when they fall in there. And if you have a larger effective population size, you should be able to purge deleterious variation faster. But by that logic, there should be fewer of those in the fish because fish have enormous effective population size, but we don't see that. But I'll show you another result about that too. But the other thing is that's cool, super crazy about this is the outliers. So here's like this weird outlier here. This is another weird outlier here. And this really wild one here is in the fish. You see this see the fish get like really big, like peaks get really high there. So it turns out all of these fish that have like this largest number, I have it here, these guys here, all of these, turns out if you look at these are all from this same massive clade called lantern fishes. So these lantern fishes, which are these fish that glow in the dark underwater, are basically have more structural variation diversity than any other species on Earth. And perhaps not, perhaps not, not too, maybe, I don't know about not surprisingly, but very intriguingly, this is like the most abundant taxa by mass of vertebrates on the planet. So this is like an insanely diverse group of fish.
So I'm going to claim that I can't remember what you're going to say next because does the larger effective population size give more opportunities for non-allelic homologs recombination?
I hope that would be, interesting. Yeah.
So the diversity is like encouraging is the argument that, now you have more differences between the copies. Now that means there's more opportunity to make new
Maybe.
copy number states.
Yeah. I hope that's the case. There's one case, the case that I actually think is happening is a little bit simpler and more boring, but it's actually really cool too. So we'll get there. My hope, yes, but you'll see in a second. Yeah. So here's another way to look at this data. So this is a histogram, believe it or not, turned on its side. So each stripe here is a heat map histogram. So just look at the top one here. So ignore this bottom one. This is the proportion. This is the count. So basically hot, or basically blue means hot, more, and yellow means less. And then this is going from small things to big things down here. And so what you're seeing is the size distribution, kind of similar to what I showed in the chimpanzees, right? And what you start to see here is you start to see signatures popping out. So here, for instance, I don't know why it got cut off on the side here, but there's an axis here. Yeah, there's an axis there. Yeah. So, but this axis, these right here, this is alus. So alu elements are like super active in mammals, but they don't exist in other clades, right? And then here's something else, another sign-like element that's super active in birds here, right? And so this is and then this bottom plot is the same as this top plot, but we've weighted it now by size. So this is like how much, so you basically take this plot and you multiply it by the y-axis scale. So like bigger things will give more weight. And you can start to see other signatures coming out down here. So basically this is like a barcode of structural variation across life. And
And your high molecular weight barcode there what's the scale on that? Is that 10,000 kb? or-
this is, it goes from, that's a great question. It goes from 100 at the top to 10,000 at the bottom. Yeah. So this is here, this is like 1,000 or 3,000, right? So so it turns out this is the L1. This is the line element.
Yeah, 7 kb, yeah, you're close to 10 to the 4, which is 10,000.
Yeah.
100,000.
Oh, yeah.
Yes,
Yes. I was off by an order of magnitude. Yeah. Yeah.
No, that's the line, yeah, that's the line, yeah.
All right. So what are these? Well, you already know a lot of these are mobile elements, and we can figure out what they are, and we can count them. So we can make another plot, very similar plot, but where we label what kinds of mobile elements these are. So the top here are class 1 transposable elements. So class 1 transposable elements are copy paste transposable elements. And so these are like, so here again, here's our alus here. And of course, I can't even read this. I can even read this. No, this is line. Sorry, this is the line peak here. This is a massive bunch of lines here. And over here these are LTRs in lizards. And so there's a huge expansion of LTRs in lizards. And yes. Oh, is it the count? Yes, it's the count, but it's been normalized to the size. Yeah. But you can see all of this TE activity happening in the lizards here. And okay, and then here's the class 2 elements. So class 2 elements are really interesting. Class 2 are cut and paste elements. And these have been entirely silenced in mammals. There are no more cut and paste elements with one exception here. Does anyone know what the exception is for DNA transposons? You can read it. It's in bats. So bats have an activation of these again, right in here. Yeah.
What's the very bottom line, sorry?
The bottom ones are low complexity repeats. So these are like simple things here. Right?
Oh, the bottom one says non-repeat. Does that mean that it's like stuff you couldn't classify in general, like all the other stuff?
So yes. Non-repeat, it doesn't, okay, this needs to be updated. What it means is we couldn't assign it to a class.
Yeah, basically.
Yeah.
But it is embedded in
No,
It's
sign other Y, it's only active in the primate lineage, apparently.
That's a line now. No, it's short.
This is the lines, this is the signs down here.
Those are the other Ys.
Yeah, these are the signs right here. Yeah.
Okay.
So what you can do is you can then go back to this plot and you can start zooming in to try to find like weird stuff that's happening. And so here's a case. So here's our bats. Okay. And I've zoomed in on the bats. And then here's this size distribution here. I'm doing for time. Doing fine. So here's the size distribution here, which is super cool. And you can see in these closely related bat species, there's all this different stuff happening. And what is this? So this plot here is the same if it was, I know these are a bit confusing, but it's the same as these plots. It's just in a heat map form. So here's size and count on the y-axis. And what you're seeing here happening is, let me see if okay. So in the ancestor, there was this massive activity of line 1s that is still ongoing, okay? But in these subclades, that line 1 element was silenced and it got turned off because in these subclades, you don't see any more. And in turn, this unknown element got turned on. It's probably some sort of sign element and has been then sort of gone crazy and been co-opted in these other bats. Here's another case in these really beautiful jays. And
But you showed it is the same repetitive element. right?
this one here. Yeah, we don't, yeah. And it's not, yeah. Yeah, the problem is that we don't know. It's really hard to annotate repeats. So we should, but what we need to do is we need to build, the way this works is we build models from every genome individually using a repeat modeler. And then the repeat modeler has these basically these HMMs, and then we go back and we try to figure out which one was which, but it sometimes fails. So in some of these cases, and so we're basically trying to make it work better.
One type of thing or more than one?
No, lots of different things, lots of different things in the unknown. Yeah. But they're not like, if we were to, if you were to sit down with like NCBI and play around, we could figure it out. It's just that we're trying to do this at scale, right? Here's another case in these birds. I don't know what's going on here. Basically, yeah, this class gets activated in this lineage, and it's still active here, but then it's got silenced in this lineage, and it just popped up. And this is a crazy example. So this is in whales, right? And so here's these whale species here, and this is wild. So look at the y-axis scale here. So this is a hippopotamus. There's like 2,000 of these unknown elements. We'll figure out what they are eventually. And then in this Mesoplodon incerotris, or whatever it is, there's 30,000. So you can't even see this axis here. It's just exploded, right? And then eventually you got silenced in these subplates here. But there's just insane, in these weird whale things, there's been insane explosion of these mobile elements. And again, this is contributing, this is not just like in the reference genome, this is segregating in the population. So what that means is that these are like active now. These are happening now because they're heterozygous. So all kinds of stuff. Yeah, we have two chromosomes, right? We just have hap1 and hap2. So we only have two, we've drawn two chromosomes from the population, and we're inferring everything about them just by looking at those two chromosomes. Yeah. Okay. So this question about that Enza you brought up, and Eric you as well, thinking about effective population size and your ability to purge stuff. So we've been exploring this a little bit, and you can take structural variants, and you can classify them and SNPs, and you can basically classify how deleterious they're going to be. So we did that, and so here's like SNVs, indels, and SVs classified by high, moderate, and low impact. And kind of with your expectation, there's actually more. The load is not the right term, but there's basically more deleterious variants in the ray fin fishes, probably because they have a higher effective population size. And so you can see that by what you can do is you can plot the heterozygosity on the x-axis, and on the y-axis, you plot the heterozygosity of the high impact variants.
And higher numbers, ray fin fish have higher loads, deleterious variants. That doesn't resonate with having a high effective population size, because they should be able to.
Yeah, load is the wrong term. They just have higher numbers, and they just have higher numbers. And I'll show you why here. Basically, yeah.
So are you considering like some species basically are R versus K? They have a lot of offspring, but only some survive versus they have few offspring that survive.
Not directly, but that's sort of what you're seeing here. Basically, high heterozygosity species are going to be those. I always get mixed up which one is which, but those are going to be those live fast, die young. So fish are often like that. Basically, have a lot of offspring, right? Tons and tons and tons of offspring, and there's tons and tons of them. Versus like mammals, right? And so you're kind of seeing that here. So here, what this shows, I mean, there's too much data here, but let's just take one example. Let's just only look at mammals. And also forget the indels. The indels are above. But if you look at sort of the proportion of high impact variants on the y-axis and the heterozygosity on the x-axis, they correlate. So what that means is that, and this has been seen in African human populations as well, more diverse populations have more deleterious variants, but they just have more variants overall. So it doesn't mean that the load, you're exactly right, Rob, is the entirely wrong word. Like I shouldn't have said load. They just have more. But what we can then do is we can look at the proportion of those as a function of all impact, of all variants of significance, all variants that have impact. So this is the proportion of high impact variants out of all variants that impact things. And this actually declines with heterozygosity. And so what that says is that there is more deleterious variants overall in these big populations like the fish, but the bigger the population, the better they are at being able to purge these things, right? But there doesn't really seem to be a difference in any of these between structural variants and SNPs. Yeah. Okay, so the last thing I'll talk about with this, which I think is super cool, and it comes back to when I was telling you about it, this comes back to this analysis I told you about at the very beginning, where we were looking at mitochondrial genomes and somatic mutations, and it was different in humans and chimps, or in humans and mice. And we saw this a couple of years ago when we looked in our goofy fish too. And we did this analysis where we looked at segregating genetic variation. So SNPs in the population of these rockfish. Some of these rockfish live for 10 years, and again, some of them live for 200 years. And I had this hypothesis. So one of the way these methylation clocks work, or the number, or the kinds of somatic mutations you get with age, are these cytosine deamination events, CpG to TpG events. So that's basically your signature of aging. Okay, and I had this hypothesis where if you have a fish that has a generation time that's like way longer, or a much longer lifespan than another fish, then the types of mutations that are going to be in that are in the sperm and eggs of those much longer lived fish should be different, because some of these mutations occur as a function of age. And we indeed saw that, right? So the lifespan correlated with the proportion of variants in the population that were CpG to TpG, which I thought was kind of mind-blowing. But what that means is that there's potentially differences in the mutation spectrum in the segregating variation across all kinds of different species, maybe for reasons like how long you live and your body size, but maybe other reasons too, like you're better at DNA repair or something like that, or you've adapted to flight and redox reactions or something like that. Are we going to say something, or you?
So the DNA repair you have to worry about as a soma is all mitotically driven rather than meiotically driven.
Mm-hmm.
So our brains are post-mitotic for the large part, and yet we still brilliantly accumulate a lot of mutations.
Yeah.
So I guess I don't quite understand the advantage of going. Well, we'll come back to it.
Okay. Yeah. No, I see where your head is going. And there's a lot of ways your head could go with this, because it is kind of weird. And we also are kind of playing fast and loose with mutation, right? I was talking about somatic before, and now I'm talking about germline mutations. But the point is that there's differences in the mutational spectra between different species, it would appear, both at the somatic and the germline level. So we did this analysis. It's kind of a pain in the ass, but it's really cool. Basically, you take these genome assemblies and you pick a focal species, and you find then its closest relative, and you use that as its outgroup. And then you align that genome to its outgroup to figure out the ancestral state of every single SNP that's in the population. So you're taking SNPs and you're trying to figure out what their ancestral state is. And so you align them to the closest thing in the tree. Now there's going to be biases with these, right? Because some of them will have an ancestor that's further back, et cetera, all these things, but it kind of works. And then we make a pseudo ancestral genome, and we estimate the mutational spectrum. And we get this plot that looks like this, which is super mind-blowing and super cool. All right, so what is this showing here? This figure here is showing the same thing as here, but it's showing it for everyone. So what is this? This is the proportion of A to C's in all contexts. So this is TAA to TCA would be the things that are happening here. So these are all trinucleotide contexts of all different mutations. And so here we have this as well. Here's all C to T mutations in all trinucleotide contexts, all C to G, C to A, A to blah, blah, blah. And if you squint, you can start to see there's like patterns here, right? There's different stuff happening. Of course, you really have to squint. So let's zoom in on this thing, right? So what you can do is you can take this thing, this monstrosity, which is basically just a table, and you could do PCA on it. And when you do PCA, you start to pull apart clusters of signatures. And so here we did PCA on the mutational signature of SNPs found in the population. Here's PC1 and here's PC2 here. And you start to see, remarkably, the fish are separating from everybody else along PC1 axis. And the PC2 axis seems to be driven by CPG to TPG mutations. So basically across clades, there's different signatures that you're seeing. You keep going further down here, and you can actually break things apart even more. So who is this? This is just, okay, who is this? No, this is just the mammals. So this is just the mammals here. Okay, so you might say that that's like the crappiest PCA I've ever seen, Peter. And fine. But actually, if you do, it's kind of crazy, right? You see rodents are all on this side of the PCA, and other, who are these blue guys? I don't know. Whoever that is. What is that? No, what is an artidogactyl? I don't know. Like an anteater or something? Anyway, the point is that there's stuff happening here. There seems to be biases and mutation that are happening between different species, which we had hints for when we looked at the somatic data. And now we think that it's different, like this is happening in the population as well. And here's a really cool one. So here's again, also, I think just mammals and PC4 versus PC3. And up here in green are all the bats, right? So the bats have their own mutational signature. And so now we're trying to extract and figure out which mutational signatures are coming from which species. The other thing we can do, which is really exciting, is we can look at genes that get hit by structural variants, and genes that get hit by structural variants and are structural variants that are segregating in the population that have happened repeatedly throughout evolution. So they've happened in one clade of fishes as well as some clade of birds. So these are genes that are segregated. They're not just being lost, killed, but they're being copy number polymorphic across individuals in the population in multiple different species. So, for instance, this is the mucin genes. Mucin genes are highly polymorphic, this gene mucin 2, in ray-finned fishes and a little bit in birds. But basically, in different fish, mucin genes are being repeatedly hit by structural variation. And that's probably because these mucins are involved in immune defense in aquatic environments. And there's all kinds of crazy. I mean, this is again just brand new, ugly looking data, but I think it's super cool. Olfactory receptors, if that makes sense. And I'm trying to see. Yeah, there's another one here, and I can't remember. This one is something too. Is there something? There's something. I don't remember the difference between these two plots. Yeah. Again, a bunch of olfactory receptors in birds. So birds seem to have a lot of turnover of these olfactory receptor genes, which is interesting. That's the same one. So anyway, so that's conclusion of this part, which is extensive clade-specific variation in SV and SNV diversity, which I think is super cool.
So if you go back 3 slides to, so if you were to plot maximum longevity per species on this, it would be everybody who's an elephant is on this side and everybody who's a small mammal is on that side.
Yes. Yeah. Which I want to do that. I want to basically go systematically correlate longevity with each of those. We haven't had time. This is all brand new.
You said PC1 was CP and CPG.
Yeah, PC1 looks like CP, or PC2 actually, it looks like CPGs. No, PC1 is this. I have one concern. I think I'm a little worried that PC1 is a sequencing artifact. I don't know this, but we have to follow up on this because that PacBio has these issues apparently with high GC content. And so I wonder if there's a bug here that is happening with, and these fish genomes tend to be more like that. I don't think so because the amphibians are here too. It doesn't really make sense to me, but it's a worthwhile thing to explore to make sure that it's not a bug. PC2 is the CPG. So this one is probably very strongly correlated with lifespan, the y-axis here. Okay, so that's that. And then, okay, I'll tell you, I have like 5 slides on this other topic, on this crazy other idea that we just started working on. So, but we don't have to do it. Okay, everyone doing okay?
Yep.
Any questions? Okay, so that's then finished project number 2. So, and this one is really rough. Okay, so structural variants are cool. Hopefully that's been established. And when you look across the genome, they are not always happening, they're not occurring in a random way. So when you look, this is structural variants in humans. And this is just the ideogram with showing the sort of the number of structural variants. And you'll see numerous hot spots, right? Like there's one on chromosome 8 here. They tend to be happening in the subtelomeric regions, right? And then this is not surprising. But why is that? That's the case because there's both sequence features and biological mechanisms that result in lots of SVs at those regions of the genome. At the same, and you could start to think about these. This is this insane plot from this paper on bioarchive, Schleusnig et al. From Tobias Marshall's group. This is basically just huge set of structural variants from humans. And this nasty, horrific plot here is showing SV length for insertions and deletions. And then they basically went and they tried to find homology outside the structural variant to see what was there homology on either side of where the SV was. And then this is plotting the length of that homology. So this huge cloud here is ALUs. So what that means is that there's a lot of structural variants that are created by recombination between two ALUs that are separated apart, and then they basically recombine with each other, and then cuts out a piece of the genome. So that's what that's showing. But the only reason I want to illustrate that is that there's signatures in the genome of where structural variants should occur, right? So there was this undergrad who came to our lab. His name is Davin. And he's this genius kid from Singapore, and he was really into using machine learning and to exploring machine learning models. And over the last little while, we've been playing with these models, and this is the model that we've settled on. I should be explaining this a bit better. What the task is we basically take a window of the genome, maybe it's 5 kb, or we've tried different windows, and we center it on a structural variant that exists. And then what we do is we try to predict if we get a whole list of the ones where we know that there are structural variants that occur there, and a whole list of them where we know that where they're of control regions. And then we try to build a classifier that can predict, is there going to be a structural variant in this region? And we've tried this in a bunch of different ways. And this is the model that has ultimately shown the best results. And it's very, very strongly influenced by Splice AI. So Splice AI is a model that predicts, basically it predicts intron-exon boundaries. And so it's kind of like a gene prediction tool. But the way it works is you take your input sequence, in this case, like 10,000 base pairs, and you pass it in series through a bunch of these nested convolutional neural networks, or convolution layers, right? And so these different convolution layers are sort of looking for, they're looking for motifs, essentially, over and over and over again throughout this, right? It's kind of like a position weight matrix on steroids, all multiplied by everything. And then we added this other part here. We stuck this other little thing in here, which is 22 genomic annotations here. So this is a self-contained model here, which will predict if you have a structural variant. And then we can make an additional prediction based on putting in annotations here. And so there's two things. We can basically have the output from this model and then the output from the combined model together. And this thing works like preposterously well. So here's just the neural net only, and different window sizes, right? So let's look at 400 base pair window size, for instance. And this is the area, this is the like a receiver, a rock curve, the area under the rock curve. And then this is this is the area under the rock curve, and then this is the area under the precision recall curve. So basically these numbers you want to be as close to 1 as possible. And you're getting like 0.92 or like 0.9 at these best things for being able to basically take a random piece of sequence and predict if it's going to have a structural variant in it, which is wild. And you can improve this to like 97 or 98% accuracy if you stick in genomic annotations. So what are these genomic annotations? You can see here they are. So, you know, GC content, distance to nearest ALU, distance to nearest line, sign, things like that. then here's the CNN, what it's predicting, and then this is just showing the correlation between these features. So the CNN is actually not really correlated that much with things, but it is correlated with the number of SNPs, and it's sort of negatively correlated with philo P score. So what that means is that with no information except the sequence, you're able to predict philo P score, like no alignments to anything. You're getting a predictive you're getting like a pretty good correlation with philo P score, and you're also predicting regions of the genome that are highly mutable because they're getting SNPs, which is pretty crazy. And so the other things that
Yeah, there's this kind of question here. It's like, so DNA's overlaps.
yeah, this stuff, okay, so basically
Encode.
all
like,
of this is the encode crap pile. So we need to reduce it down because these are like number of base pairs with cis regulatory element overlap and average. These are like the same. So we should just kill one of those and these other things, but that gives
You just think they're correlated.
they're just correlated, yeah. So we should get rid of those, yeah.
And then you have another, well, it's interesting that you have these two blocks. The other block is the D content.
Yeah, but like, so this model. Right, so you can do, so this is what this looks like. Remember that there's two parts to this model, and the CNN works like really well on its own. It's like 90%, and then you can get that extra bump by adding in the annotations. And this is what the importance of those annotations is. So the strongest predictor on top of sequence of like, if you're gonna have a structural variant is if you have other structural variants nearby, or if you have other SNPs nearby, or small indels, and then find, then philo P score, yeah. And then, you come down, recombination rate is down here. Simple repeats, yeah. But, okay, so here's the thing that's really crazy though. So the reason why, the thing that's interesting though is like, so these things are weird and we don't know what they're doing, right? But one thing you can do, here's what I thought. I thought when we built this model that this model would be basically predicting GC content or something. But it's not. Like this CNN is not correlated with GC content at all. It's like, or maybe a tiny little bit, right? Right. But so it's on its own, it's sort of discovering all these things about the genome, about where structural variants are. And so we can, so we're basically what we're doing now is we're exploring this model to try to figure out what it does, what's under the hood here. And then we can take this. So like this plot here I think is pretty cool. And then we can do this again across, we have all these different fish and all this crap here, and we can start to predict structural variants in different species. And that the whole, why the hell would you want to do that? The goal behind doing that is that you can, learn, are there different things that are driving structural variants in the ray fin fishes versus humans? And is that why they have higher loads of structural variants?
Go back one slide. What about CPG islands? So I see G.
Yeah, that's a good point. Yeah, we didn't add that as a feature. Maybe we should add that. Yeah, and I don't have it on here, but so what we've also extracted, we haven't done it yet, but we can extract from this. We can extract the k-mers that are coming up, and the k-mers that are there are sequence features that are strongly predictive of structural variants, and not surprisingly, they are k-mers that are enriched in DNA structures, non-canonical DNA structures. So G quadruplex forming structures. We haven't done the Z one, but probably, right? Yeah. So I just think that's kind of cool. So this is, yeah. Anyway, that's it. That's all the stuff I have. I've got tons of stuff. And yeah, so super great thanks to the lab, and particularly again to all the folks here who we've been working with on all this kind of stuff. And thanks for all the great questions throughout. Yeah, how?
So how do we detect structural
Yeah, good question. We did it by, there's a lot of ways. It depends. It depends on which project you're talking about. But in the project where we looked across the diversity of species, we used basically I think we used FIMASM, which is a tool that takes two structures takes two haplotypes that are aligned and tries to identify differences between them. It's kind of like sort of like a pangenome approach, but not a pangenome, right? The other way to do it is you build a pangenome and you look for bubbles between the two, which would be a great idea, but the swimasm was a little bit more well-behaved.
Well, for multi-sample stuff, it helps to get you. But
Oh, yeah, we only have
just-
two. We only have two, so it's super, yeah, so it's easy.
Yeah. It's just a barrel.
We can talk, oh, yeah.
No, about the last.
When you compare the structural variant between different clades,
Hmm.
how do you manage possible batch effects or different qualities? of
Yeah.
assemblies?
So what we did is we filtered for we're basically only hi-fi assemblies. And they have to be, they have to have certain quality metrics. And we did this analysis, like you can do this analysis where you can look at the total number of structural variants as a function of genome size, and it's not correlated and things like that. So we did a bunch of those things. And then we have kind of a hack, which is that there's no systematic difference in the genomes from the different clades. So it'd be very disturbing if like all the fish were done first and then on the other species, but they're not. They're coming from all over the place. Yeah. And then we have a third hack, which we have like a bunch of gold genomes where you can just like only look at the gold genomes. And we know we've like very carefully vetted those ones, and they look very, very good. And so you don't really see huge differences, but there is probably at the clade level, I don't think it's causing anything. There could be just certainly one-offs. We found things. What the biggest error we found so far is people who have uploaded the same haplotype twice. And you're like, this cannot possibly be true. This is not a diploid. Yeah. That happened a lot for the Canadian Genome Project, it would appear. Yeah.
So how do you define a gene, Peter? Because after this kind of treatment in your introduction, that it's really the haplotype that is often a selected unit.
Yeah.
It has to stay together. And so, Dawkins says a gene isn't what we think of as a protein. It's a bunch of DNA that hangs together over many generations of recombination.
Yeah, that's a good question. We just thought about it in the simplest sense that it can possibly be, by just looking for coding regions. But basically, all of these, all those mobile elements, those are genes. Those are things that are, they're.
You pointed out that MHC has been hung together now for, did you say 60 million years?
There has been some recombination.
There has been some recombination, yeah. I mean, I don't know. I think.
It's effectively acting like one gene. It's just not one protein code.
Yeah, because it's one. Yeah, sometimes people call those super genes.
But so the structural variants, your whole presentation, you started with the word resilience, genome resilience. Never really came back to that.
Oh yeah, because that was a slide from a different talk.
You told us about genome flexibility.
Yeah, but not
We
resilience.
don't know why. So I think Kevin pointed out R versus K.
Yes. Yeah.
That's a big deal. Fluctuating climate,
Yes.
big deal. Fluctuating population size, big deal.
Yeah.
So when you put it in a true evolutionary ecological context, then you kind of can see the advantage of structural variants, like in hummingbirds, where the mutation in one of the actins lets them flap their wings super fast.
Yeah. Yeah.
Right? And that mutation turns out to have been a huge adaptive advantage for that entire clade.
Amylase, which you didn't present, but some folks here probably heard about it, but that's an example of the resilience in some sense, because you have this rapid selection. Sorry, you have this rapid evolution of types that then you select.
It's almost group selection, isn't it?
It is kind of, yeah. I was asked to go talk at like a resilience seminar, so I called it resilience.
Oh, okay.
I just thought that was my intro, but see the point. It's like, we don't really come back.
Question on the AI part. So.
Me too.
You show different resolutions, right? With the lower resolution, with the small scale, 100 BP, it predicts very well, but when you go up to higher KB.
Oh, yeah. The reason why is, so the reason why is that these take more and more memory to train. So we haven't done 5 KB yet.
So my question is, are you predicting the breakpoint or are you predicting the entire structural variant?
Yeah. What we're doing is actually kind of lame. We're saying in this 5 kb window, there is a structural variant. There will be somewhere in there. But we actually have the probability on a single base level. So what we did, because you can slide around it. So what actually you can do is you can see, I don't have a picture of it. I'm trying to make a meta plot of this across all known structural variants, but you can actually see it kind of like peaks. It doesn't peak right in the middle, it peaks like right beside, but the probability is kind of like it is working. It is like zooming in on where these SVs happen. And I had this, so I think we're not going to play with it. We're just going to look for this. But in my mind, I was like, yeah, because you could slide along the genome and look for homologies, right? And that if you find like pairs of homologies, those will be hot spots for, recombination and SVs happening, right?
But if you won't train it on the exact breakpoint, then the thing should always be right in the middle.
Oh yeah. So well, yeah, you get technical artifacts if you do that. So we wiggle it around a little bit, but yes, it should be in the exact middle. Yes. Well, I don't know. What's the question there?
So I'm thinking if you starting from the training, you put the breakpoint of the structural variant right in the middle of the window.
Yeah.
Then it shouldn't matter where the how big the window size is again, because they are all the size of structural variant.
Yeah, I don't. Yeah. Why is it that when you get when we have more spacing, it doesn't do as well? I don't know. It could be a training aspect, and it could have to do with the size, because these convolution layers, you could probably get the same performance if you tweak these convolution layers for the different sizes. And that's what they did in Splice AI. But the problem is that, so, okay, here's the real thing, is that the bigger your size gets, the more of these layers you need. And it gets more and more hard to train. And then so what was it called? There is some way that in some of these giant models, what they do is they use dilation. And what dilation means is just sampling. So like you have your convolutional part is like a huge window, say like 10 kb wide, but you only take every 5th base pair or something. It's like all these words in machine learning that are just like not complicated, but they make it sound very complicated.
Is that sparse training?
Yeah. Andrea.
Can you predict also when and how they will happen?
Not.
Estimate.
Well, so okay, there's a bunch of stuff. This is brand new, and it's all like there's a lot of stuff swept under the rug here. So no, but well, I don't know. So what I want to do is I want to look at these numbers as a function of allele frequency. I have no idea, and I'd be very curious. My guess is that they will. I don't know. I actually don't know what to expect if they're going to work better for things that are at high or low frequency. My suspicion is that they'll maybe work better for things at low frequency, but I don't know.
Vision transformer.
Okay.
It sort of bridges between the convolutional neural networks and long sequences. So maybe vision transformer could be something.
Okay. Yeah, that's a cool idea.
This will apply to multiple spaces.
Yeah, yes, we did that. So we have run this on a bunch of different species, but we've only tested and trained on the same species. Like you can learn weird things, I suppose, if you like plug one model into the other species. So that'd be kind of fun.
You could learn across all of them. That'd be interesting.
Yeah, we thought about that, but yeah, yeah.
What are the common features? Like then you ask me more about like what is the nature of the evolution in general?
Yeah.
Is it basically validated in these?
Yeah,
That's so cool, right? Just to do the prediction and then.
Yeah, I thought about trying to train it in like a chimpanzee and then test it in a human or vice versa. I don't know. Yeah.
So does the X chromosome behave the same or differently?
Oh, that's a good question. We haven't looked yet. I haven't looked specifically at that.
I'm not a geneticist the way you guys are, but in terms of like starting from the beginning, so cell cycle is core. you take genes that are all involved in the original cell cycle, and you have layers with evolution added on. Is the dispersion of those genes sort of random throughout the genome, or is there?
Oh, yeah, that's kind of like, yeah, that's kind of like, yeah. They tend to be quite distributed for the most part, yeah. Cool. Thank you, everyone. I appreciate it.
Retired.
Yeah.
Maybe we could discuss
Yeah,
with-
don't get around.
Yeah, we can go talk.
do this.
Sounds good. Thank you so much, everyone. Timing.